Photosynthesis is the process used by plants to turn light energy into chemical energy that can be used for other activities within the organism. This is shown by the reaction of carbon dioxide and water, which yields glucose and oxygen. This reaction occurs in the chloroplast of a plant cell. Photosynthesis is divided into two parts, the light reaction portion and the Calvin cycle. Together, these two processes work to help the plant make sugar it can later use in cellular respiration. The light reactions begin in photosystem 2, where a photon of light strikes a chlorophyll molecule, exciting its electron to a higher energy level. As this electron falls down from this state, it releases a photon of electromagnetic energy, exciting an electron into a nearby chlorophyll molecule. This pattern continues until one of them reaches the pair of chlorophyll A molecules. At this point, the electron will rise into the electron acceptor and pass through an electron transport chain, a series of molecules that accept or donate electrons easily.
The electron chain delivers the electron into photosystem 1. In a similar fashion to what occurred in photosystem 2, light is received and excites an electron that bounces around until it finds a pair of chlorophyll molecules. When it reaches this pair, it rises until it reaches the electron acceptor and leaves a hole in the pair of chlorophylls. This is where the electron from the chain comes in and fills the hole left by it and eventually meets the other electron in the acceptor, where they will travel through a second electron chain.
Through this chain, electrons are moved step by step in a specific direction across the membrane, eventually being paired with NADP+ to make NADPH, which will eventually be used in the Calvin cycle. Meanwhile, hydrogen ions are also being transported through the thylakoid membrane through the breakdown of water. This sets up a concentration gradient within the thylakoid lumen, which will be utilized by the enzyme ATP synthase. Because of the high concentration of hydrogen ions inside the thylakoid, ATP will want to pump hydrogen ions out into the stroma in an attempt to re-establish equilibrium. When ATP synthase accepts the hydrogen ion, the enzyme will change shape and turn, which facilitates the creation of ATP from ADP and an inorganic phosphate group. Together, the NADPH and ATP travel into the stoma to be used in the Calvin cycle. The Calvin cycle is where sugar is synthesized that will eventually be turned into glucose. The Calvin cycle occurs in three basic parts: fixation, reduction, and regeneration. Fixation begins when carbon dioxide enters through the stomata and diffuses into the stroma, where it binds to RUBP, a 5-carbon chain with phosphates at either end, and is catalyzed by rubisco. This reaction forms a 6-carbon chain that instantly becomes 3 carbon chains, called phosphoglycerate. This is known as fixation because the inorganic CO2 is fixed into organic compounds. Reduction is when these 3 PGA become converted by the ATP and ADPH's stored energy into G3P. First, the ATP is broken down into ADP and the phosphate group, which, using the energy released from the splitting of ATP, is attached to the 3 PGA, creating 6-biphosphoglycerate. NADPH then donates an electron, breaking off an inorganic phosphate group from the biphosphoglycerate, creating 6G3P. 1 G3P leaves the Calvin cycle to contribute to the formation of the glucose molecule. Meanwhile, the others become RUBP for the continuation of the Calvin cycle, also known as regeneration. Once there have been 6 cycles of the Calvin cycle, 1 glucose is produced.